The purpose of a scientific investigation or experiment is to gather evidence. The evidence is in the form of data and observations, which are then used to evaluate a hypothesis. And hypotheses are generally developed to test one part of a theory so that we can understand the how and why of natural phenomena. Hypotheses are narrowly focused on one small part of the phenomenon and predictions of what is expected to happen, which are based on prior knowledge and experience. Theories are generally based on many pieces of evidence from hypotheses, making them more general explanations and predictions. So how is evidence gathered? A well-designed experiment is required to collect reliable data that can be used to evaluate a hypothesis. This is where the scientific method comes in again. After observations, the first thing that needs to occur in designing an experiment is asking a relevant and fully focused research question that is clearly described. This is the case whether you are developing your internal assessment or extended essay in the experimental sciences. What's the difference between a good and bad question? Let's say you want to investigate plant mineral nutrition and how growth is affected. A bad question would be, what is the effect of fertilizer on plant growth? A good one could be, how does a sulfur deficiency affect growth of Helianthus annus? The first question is far too general and not repeatable. Additionally, it neither shows interest or significance in what is being studied, nor does it have a real answer. Sure, fertilizers help plants grow, but to what extent? The second question is much better. It actually allows for making informed decisions about the rest of the design of the experiment. It allows for creating a falsifiable hypothesis and determining what data will be collected and how. This takes a lot of practice, but in due time you can begin to ask really good questions. This brings us to our focus, the Meselson and Stahl experiment in 1958. It is one of the significant experiments that grew from Watson and Crick's elucidation of the structure of DNA. With Meselson and Stahl, it was very much inspired by the Hershey and Chase experiment, which provided evidence that DNA and not protein is the genetic material. One function of living things is reproduction. This is the production of offspring through passing on heritable traits. This could be through asexual or sexual reproductive methods. In this way, DNA must serve two functions. One is to store and express heritable traits. The other is to replicate itself so that it can be passed on to new generations of cells that will retain those heritable characteristics. Based on these functions, Meselson and Stahl began by asking how DNA is replicated. To design their experiment, they needed to have an understanding of the composition of DNA. DNA is composed of pentose sugars, 5' phosphates, and 1' nitrogenous bases. Early experiments and assays verified that DNA contains nitrogen. The most common isotope of nitrogen is N14, representing over 99% of the isotopes of nitrogen. N15 is a distant second, but is functional and stable when it is constituted in DNA. N15 is heavier than N14 and can be separated within a suspension fluid through centrifugation. If they could culture a population of organisms within different media containing one isotope of nitrogen each, they could determine through evidence how DNA is replicated. They selected E. coli as the model organism and began by culturing it using N15. As expected, E. coli reproduced by binary fission. To reproduce, the parent bacterium needed to replicate its naked circular chromosome. Since N15 was the only building block available for its nucleotides, after several generations, all E. coli possessed N15 containing nucleic acids. This was established via centrifugation, which produced a band of DNA in a specific region of the vessel. They then removed some of the E. coli and cultured them in a medium containing N14. Depending on the environmental conditions, E. coli reproduces a new generation every 30 minutes to 1 hour. Meselson and Stahl removed samples from the medium at specific intervals and repeated the process of centrifugation. The process of centrifugation produced bands of DNA of varying thicknesses at specific density levels, indicating the proportion of N14 to N15 within DNA itself. A pattern emerged from the results, allowing them to suggest that DNA replication is semi-conservative, in which one complete strand of each double helix is derived from the parent molecule. Meselson and Stahl identified a phenomenon that hadn't been explained through the support of evidence. They began with asking a clear, focused research question. Then, they developed a falsifiable hypothesis, which helped them to determine what their variables would be, and what data they could collect as evidence. They designed an experiment that produced evidence to directly support their hypothesis, and then the evidence was used to evaluate the hypothesis and support the theory of DNA replication.